Okay, it sounds like we're live. So, a very warm welcome to all our participants and thank you for joining us. This Tech Talk webinar is hosted by APMEN in collaboration with the Outreach Network for Gene Drive Research. And in that regard, I'd like to thank Tamsin Hibbert and Joyce Najum for your sustained partnership from the Outreach Network. I'm going to ask our APMEN Vector Control Working Group Chairman, Dr. Mohammed Mukhtar, to also say hello and just add a few words maybe about APMEN. If you don't, Dr. Mukhtar, over to you. Uh, Dr. Mukhtar, I'm not sure if you're on mute.
I don't see Dr. Mukhtar is here, so I think he just have internet connection.
Okay. All right. So let me just then say quickly on his behalf, because we seem to have connection problems. APMEN stands for the Asia Pacific Malaria Elimination Network.
Dr. Mukhtar
And
is here. Sorry to interrupt.
okay. Dr. Mukhtar, do you want to say hello and welcome our audience members, please, just
Thank you
briefly?
very much, and sorry for interruption. Respected colleagues, program managers, distinguished colleagues, esteemed faculty members, researchers, and students from across the Asia Pacific region and around the world. Very good morning, good afternoon, and good evening according to your time zone. It's my great pleasure to warmly welcome all of you to this APMEN Vector Control webinar entitled What is the Future of the Vector Control? I'm Dr. Mohammad Mukhtar. I'm Director National Malaria Control Program Pakistan, and I'm also Chair of the APMEN Vector Control Working Group. To keep my introduction brief, let me simply say that APMEN stands for Asia Pacific Malaria Elimination Network. APMEN provides the technical support and promote the regional collaboration among 22 member countries across the Asia Pacific region, working collectively towards one common goal, that is the malaria elimination in this region. To support these countries, APMEN provides the support delivered primarily through 5 thematic groups: the APMEN Vector Control Working Group, which is the hosting today's webinar, the Surveillance and Response Working Group, Case Management Working Group, Climate Change Interest Group, and Outdoor Biting Interest Group. I warmly invite all of you to visit the APMEN website and learn more about our work, our regional initiatives, and most importantly, how APMEN can support and collaborate with your esteemed organization. Once again, thank you very much for joining us today. We have an exciting program ahead, and I'm confident that this webinar will provide a valuable insight, stimulate a meaningful discussion, and inspire new thinking about the future of the vector control. With that, let me once again welcome all of you, and I'm very much confident that we will have a very productive and engaging webinar. Thank you very much for all of you, and over to Dr. Liu for further proceeding of webinar. Thank you.
Thank you for those wonderful words, Dr. Mukhtar. So, as you all know, our webinar today will focus on what is the future of mosquito vector control. And we're fortunate to have 5 highly experienced global experts in their fields to share their experiences and insights on this important topic. So we're going to be focusing on several state-of-the-art, innovative, vector control tools that are at the forefront of entering and playing a bigger role and supplementing are, historic nets and indoor residual spraying. So we're going to look at some that are emerging new vector control approaches. There are 1 or 2 that, because of lack of time, I was not able to include, much to my regret. And these include attractive targeted sugar baits, which we should have included, but it's still not quite at the stage where Our other tools that will be discussed today are, so ATSBs are there in the background, but perhaps a step or two behind. So sincere thanks to each of our expert speakers for joining us today. Some quick housekeeping issues. Please note that the webinar is being recorded and it will be made available after conclusion of the event on the URL, which I think Wulan will place in the chat box. To avoid background accidental distracting noises, we also will mute our audience members and disable video mode. But I'd like to very strongly encourage our audience members to pose questions to our speakers. And these questions will be responded to during the Q&A sessions. So here's an important little snippet, please, and I'm asking you to adhere to it if you don't mind. Please put your questions in the Q&A box, not the chat box, please. There's a lot of clutter going on in the chat box, and it's difficult to extract the questions from there. So if you have a question you'd like to pose, put it in the Q&A box, please. We will not be monitoring chat box for questions. Also a suggestion, please put the name of the particular speaker you would like to respond to your question in front of your question. For example, Carlos, please can you briefly explain how one can access the particular product you were talking about? Please note that all the presentations will be given back to back, and then we move on to the Q&A session. Right at the end of the webinar, we will also have a very short multiple choice poll survey questionnaire so that you can indicate your level of satisfaction regarding the event. Okay, so let's move on to our first presentation, which happens to be mine. And it's just a broad discussion, introduction, providing context to the presentations that will follow. Wulan, may I ask if you could share screen my presentation? I seem to be having problems with the...
Sure, will do.
Thank you so much. Okay, so the loose topic, it's a loose hat for some general discussion. Next slide, please, Wulan. And I'm going to be focusing on malaria vector control for this short presentation, just as a illustrative discussion. But the principles remain the same for the other vector-borne diseases. But specifically malaria, in 2024, there were an estimated 282 million cases of malaria globally and an estimated 610,000 malaria-related deaths. And I mention this, you already know the statistics, but it represents an increase from the previous year. That's the important point I'm trying to make here. Next slide, please, Wulan. So in that context as well, you know, it's fascinating to me, and I'm sure to you as well, hopefully, that although malaria has been a scourge to humanity and been killing people for several thousand years, multiple thousands of years, and collectively has resulted in more deaths than all the wars of humanity throughout history. So despite thousands of years of malaria being a burden to humanity, it's only in the relative blink of an eye, just over 100 years ago, that we actually found out what causes malaria. So in 1880, Laveran found parasites in malaria patients. And, you know, that was the first understanding of what is actually causing malaria. And then 1897, of course, Sir Ronald Ross in India, he was able to inform us that it was Anopheline mosquitoes that are actually transmitting those parasites. So now we knew what was causing malaria and what was transmitting malaria. Next slide, please, Willa. So ever since that time, we have, since 1897, basically, we have overwhelmingly relied on vector control for reducing the malaria burden. During the initial period, 1900, roughly 1900 to the 1940s. It was predominantly larval source management, larviciding with oil and Paris green, draining of ditches and marshes, etc., putting screens on homes, mesh netting over windows, relocation of homes, and so on. Construction of the Panama Canal failed beforehand because we couldn't reduce malaria. Then when it became known, mosquitoes were transmitting it, vector control could be implemented, and that enabled the Panama Canal to be built. In the 1940s, all the way through to the 1990s, indoor residual spraying was introduced to supplement larval source management. And this combination of indoor residual spraying, mainly with pyrethrum and DDT, resulted in the elimination of Anopheles gambiae from northeast Brazil, elimination of Anopheles gambiae from Upper Egypt, Anopheles gambiae and Anopheles funestus elimination in South Africa, and also from the USA by 1949 using DDT for spraying and also draining and other larval source management sites. And from the 1990s, I should mention that so effective was this combination of indoor residual spraying and larviciding, together with the discovery of chloroquine and now incorporating a very potent drug for parasite control, that the whole world was now ecstatic, and here's a magic bullet combination that we can eradicate malaria. So WHO implemented the Global Malaria Eradication Program in 1955, but as I'm sure most of you will know, the combined appearance of drug resistance by the parasites to chloroquine, and also development of insecticide resistance to DDT and others, resulted in the abandonment of the GMEP in 1969. And then there was kind of lip service to the issue of elimination and eradication, and a greater focus on malaria control. Let's just keep the levels down. It's a pipe dream to aim for eradication. 1990s saw the introduction of insecticide-treated nets, another major paradigm shift. And it so effective was this that there's a publication by Samir Bhatt and al. In Nature 2015. The publication is listed over here, which showed that more than 80% of malaria cases averted in Africa, which is where about 95% of our global malaria burden is, between the years 2000 and 2015, were due to the combination of insecticide-treated nets and also IRS. So because of the use of LLINs, insecticide-treated nets, 68% of malaria cases were averted between 2000 and 2015. And IRS averted 13% of potential cases. That combined resulted in about 663 million cases being averted. Then, okay, I mention this to point out that overwhelmingly it's vector control that has been, from the discovery in 1897, it was vector control that was the main strategy to combat malaria. Highly important. Here you can see 81% of malaria cases averted was due to vector control. But then in 2015, something happened, the appearance of, yet again, insecticide resistance within the mosquitoes, changing behavior of mosquitoes and so on. So there was a plateau and we hit a wall, and that plateau still persists. Last year in August 2025, WHO published endorsement of a new a new approach to transmission reduction by way of spatial emanators. Will it work where it is needed? Time will tell. Next slide, please, Wulan. So this slide just shows the top graph, at the left-hand side of that blue line, that's 2010, and it shows you how malaria cases declined until 2015, and then there was a plateau. Had that trend persisted, you look at the green line, that's how malaria should have reduced. But because of the development of insecticide resistance, behavior change, and so on, we reached plateau, and you can see how the line is steadily increasing. Here on the right, top right, in brackets there, you'll see since 2015, malaria cases have actually increased by 22.6% up to 2024. And what this graph also tells you is it's vector control, it's hurdles and the reduction of the impact of vector control that has caused this major hiccup. And the bottom graph is simply a reflection of the top graph, and it relates to malaria mortality. Next slide, please, Wulan. So point is, we sit with a problem. Malaria control still overwhelmingly relies on vector control. With a bit of luck, maybe vaccines will bring future change, but for now, we are heavily reliant on vector control. Think of the malaria endemic world without LLINs, without bed nets, and that will tell a story in itself. We rely overwhelmingly on vector control for malaria control. But our current generation of vector control tools have reached, it seems like, it's the limit of their primary impact. And that is why we're getting that plateau effect. There are remaining aspects not being addressed with current tools, such as outdoor biting. 3 of the 4 tools that are endorsed by WHO are targeted against indoor mosquitoes. We have 0 tools that are significantly effective at scale for outdoor transmission, which is where a lot of our residual transmission is occurring. The 3 tools I'm talking about, of course, is ITNs, indoor residual spraying, and now also spatial emanators, which WHO will only endorse if it's used indoors in combination with LLINs or IRS. So it's also indoors. Next slide, please, Wulan. So what are the promising new vector control tools out there? And this is what our webinar is all about. You're going to listen to Neil and Carlos and Jackson and Claudia, who are going to talk about emerging new tools that are becoming available, proven to be effective, and they will play an increasing role in future. And then Dr. Mukhtar is going to put together, how do we approach the development of new tools. Next slide, please, Wulan. So the point I'm trying, we need another paradigm shift, such as we did from larval source management at the turn of the century, 1900s, shifting to incorporate indoor residual spraying, and then eventually also ITNs. Then we need to do integrated vector management. But to do that, we need thinking entomologists, not just effective net distributors, buying ITNs in bulk and distributing it, because that's exactly what we're doing currently. We need qualified entomologists that can actually examine and assess malaria transmission in a local context and devise or come up with locally relevant tools. It means strengthening entomological capacity, which is another layer of shortfalls we face. We simply do not have enough entomologists. So this is a quote from Ann Wilson in a 2020 publication. She said there the bold sentence: in essence, entomologists stopped being thinkers and became monolithic deployment automatons. So we need new tools, and we need to strengthen vector surveillance and control capacity. Thank you so much, everyone. I hope you enjoy the presentations. I'm going to now introduce our first speaker. Thank you for that, Wulan. Appreciate it. Our first presentation after mine is going to be by Professor Neil Lobo, who will provide us with some perspectives and findings on spatial emanators. Neil is associated with the University of Notre Dame in the USA and also the Malaria Elimination Initiative of the University of California in San Francisco. Neil's work takes him to most malaria endemic countries all over the world, and I get the impression he spends far more time in the field than he does in his home country. He travels incredibly. Neil's research aims to integrate entomology with other key aspects of transmission, such as existing interventions, epidemiology and human behavior, and also weather factors. And it's through this integration of data types and the use of modeling that his research aims to clarify how these various factors of malaria transmission interact so as to better understand the root causes of persisting transmission and improve protection on a highly granular local level. Thank you for joining us, Neil. We appreciate your time. Over to you.
Let me share my screen, and hello everyone. I hope you can see my screen.
Yeah, it looks good.
I will
Thanks.
start. Yeah. So today I am going to be talking about spatial emanators, or what they used to be called spatial repellents before. Yeah. So what is the problem? Let's define the problem. As Leo just mentioned, in spite of scale up of interventions like bed nets and IRS, we still all have malaria, right? Both these interventions work really well, but they only work under certain conditions and prevent a certain amount of transmission based on the intervention's mode of action. And most transmission we see right now happens outside these conditions, right? Where bed nets and IRS can protect. All the transmission we're seeing happens outside their modes of action. So if you can see in the figure here, your interventions on top, and where the three overlap, the humans, the vectors, and the intervention, we see protection. And when only the humans and vectors overlap, we see transmission or gaps in protection. And that's where every single case we see today comes from. So recently, WHO had this conditional recommendation for spatial emanators as a new complementary tool for malaria vector control. It is really cool that this is the first WHO recommended vector control class in over 40 years, and that's a big deal. Like, we've been doing the same thing for so long, and now we have something new. It is important to note that it's a conditional recommendation. And as Leo mentioned again, it's to be used indoors alongside existing proven methods like bed nets and IRS. So it's indoors and with bed nets or IRS right now. And that's the WHO recommendation. And I'll go over that in a second. At present, there are two spatial emanator products that have been pre-qualified and are available for countries to obtain through Global Fund funds, and this is Mosquito Shield and Guardian, and both are produced by SC Johnson. However, Mosquito Shield is not available right now. They're not making it anymore. And Guardian is available, and it's a 1 year product. It's a passive product that lasts for 1 year. So what are spatial emanators or repellents? So they used to be called spatial repellents, and I'll go over why they're now called spatial emanators. So it's a product that releases a volatile active ingredient or a volatile chemical or insecticide into the air. And this volatile chemical reduces the number and density of the mosquitoes, the vectors that can give you disease within that treated space. So they reduce human vector contact without directly requiring the mosquito to contact the product or require human behavior. They don't need the human to do anything to it every day. It's a passive product. So with Guardian, you would hang it up and forget about it for a year, and it will protect your space for that year. So you don't have to wake up and light a coil or put up your bed net or anything like that. It's a passive product, which is a good thing. So if you look at the figure below, it sort of outlines how this works. This figure was made by Sarah Moore, so thank you, Sarah. So if you look at left, the little yellow arrow that ends in red, you have a concentration that increases towards the right, right? So if imagine all the way at the right, you hang your product, there's a high concentration of the volatile active on the right. And with dissipation, with evaporation, there's less of a concentration on the left. So from all the way from the left, you have mosquitoes that approach this cloud of the compound and they get repelled. And that's why they were initially called spatial repellents. But what we found in other studies is that we have a whole bunch of other impacts. As they have increased concentration or increased exposure to the product, we have reduced entry into houses. We have mosquitoes getting completely confused and not knowing what they are doing and flying around randomly. They may land and they don't even feed. So they might land on you and they don't feed. And there's a whole bunch of other things, like they might get knocked down, don't feed for hours and hours and hours, and if the exposure is high enough, they can even die. And this is why they've been called spatial emanators instead of repellents, because we have a whole bunch of impacts outside just repellency. So, what is the current status of research and the impact? So, on the left, if you look at the spatial laminator paradigm, right, we have spatial laminators as a complement to present WHO interventions, which are bed nets and IRS. I need to put IRS in there. So, right now, that's the present recommendation, spatial laminators indoors with bed nets. And we have different kinds of studies. So we have semi-field studies, experimental hut studies, field studies, and randomized controlled trials or RCTs. And WHO requires this RCT, epidemiological evidence against the disease, for a recommendation. And at present, we have one study for Aedes and one study for Anopheles. So that's basically dengue and malaria, respectively. And that is why spatial laminators have a conditional recommendation, because we need two studies for each disease. And if we look on the right, we have implementation studies where we actually put these out into real world conditions and see the impact. And all the reds are basically not completed yet or they're ongoing. So you can see the other intervention paradigms on the left. We have spatial laminators as a standalone tool indoors and spatial laminators as a standalone tool outdoors. So just by themselves, how do they prevent disease? We have some evidence that they work pretty well against entomological endpoints only, not epidemiological endpoints, as you can see from the not yet. So we are waiting to get this data, and hopefully we'll get funding to produce this data so we can have these products recommended by WHO to function alone or outdoors. As Leo mentioned, we need good interventions outdoors. So what we see is indoor protection. We have 30 to 60% reduction in disease depending on your setting. It, of course, changes based on your mosquito species, the house construction, all these multiple things that might drive protections. We see an added benefit with bed nets, which is where the epidemiological evidence comes from. We have limited outdoor evidence. So we actually see 40 to 90% reduction in mosquito landings in entomologically. So this is just entomological evidence, and we don't see any epidemiological trials conducted yet. So we see good impact outdoors, good impact indoors entomologically, and we have good epidemiological impact indoors, but we don't have that for the outdoor implementation of spatial laminators. We see heterogeneity across sites, which is expected with different species, different conditions, with different human behaviors. You can see the lady holding up a Guardian product, and that's the one that's presently available from SC Johnson and approved by PQ, WHO. So where do spatial laminators actually add value? So if you look at the figure on the right, where humans and vectors overlap, you see a gap in protection. And that's where the mosquitoes can bite the humans and you get disease transmission. And let's say you put in bed nets, and that's where you have protection, where they all three overlap. However, spatial laminators can now function in these gaps in protection. So imagine now you put a spatial laminator there and you reduce these gaps in protection because they further reduce biting, further reduce disease transmission. And this is what the present conditional recommendation by WHO is for. Spatial laminators plus bed nets or IRS. So it can protect people when they are not under bed nets in the evenings, early mornings, and like in mobile populations. And based on the evidence, at least the entomological evidence, we should be able to protect people outdoors and in paradomestic spaces, but we do not have the epidemiological evidence for this yet. It is important to note that this complements bed nets and IRS according to WHO and should not be used to replace them by WHO because we don't have that evidence yet. And as I mentioned earlier, it's really nice that it's not user dependent. It is passive. You can just hang it up and it functions for a full year. Operational advantages is that it can protect unprotected populations, gaps in protection, basically, in basically all transmission settings. In high transmission settings, humanitarian and emergency contexts, mobile populations, and possibly outdoors based on the entomological evidence. In the picture on the right, you can see a child in Peru in his very open structure. And right up on top, the arrow, you can see Shield, which is a product, one of the products from SC Johnson, which is hanging up. You can see two Shield products hanging up there. And this was part of the randomized control trial that was conducted in Peru. Of course, an advantage of spatial emanators is that they are small and light, and so you can have multiple distribution models. For example, you can just distribute them to health facilities, or you can even might be able to package them with bed nets, right? Because that's what the WHO recommendation is for. They're small, so you can transport a lot of them much easier. And they should hopefully be cost effective, though the data from this is not out yet. I think at present, the companies that are producing spatial emanators are trying to figure out their final cost, how much they're actually going to charge for the product. And based on the implementation studies, when we actually put this out in communities and see how they function, we can look at how cost effective these are relative to bed nets and malaria cases. So it is important to note that there are remaining evidence gaps. We need epidemiological evidence for spatial emanators functioning alone indoors as well as outdoors. And this is one of the big gaps in data we have and we need. We need data on how they protect communities in implementation studies, operationally, when ministries of health distribute them. How much do they protect people? How dense do we need to have them per structure, per village to have optimal impact, to have community protection? If we use them outdoors, how does wind affect them, rain affect them, right? Does it reduce the protective efficiency? How long do they last in these conditions that are not indoors? You can imagine wind hitting the product and taking off a lot of the active ingredients. So we don't understand this as well, both indoors and outdoors, as well as in implementation context. As I mentioned before, cost effectiveness we need to figure out. Insecticide resistance, of course, as with any intervention. The second we put it up, it's a countdown towards which we see resistance. And with spatial emanators, it's not just insecticide resistance, it can be behavioral resistance because it's at concentrations. A mosquito might be exposed to the spatial emanator at concentrations below what causes death and huge amounts of selection. So there might be behavioral resistance come up as well. And of course, the last point is we need data on how to optimally deliver them in an operational setting. So in conclusion, my second last slide, they are a promising complementary tool, complementary again, because that's the WHO recommendation, particularly for addressing gaps in protection that are present with bed nets or with IRS. They can be considered as part of an integrated vector management strategy and included in vector control decision frameworks. And of course, we need more data on a whole lot of things considering this is a new intervention paradigm. We need to monitor and evaluate them in every single setting we put them in until we really understand how this functions in that specific setting. Yeah, that's about spatial emulators. And the way forward, as Leo mentioned, we are in a state where bednets and IRS have sort of plateaued. We understand how they function and we understand how they don't function. So what do we do now, right? So in my opinion, at least my thoughts, right, we need vector control. That I don't think that's going to change. Vector control is central to many diseases, and vaccine and drugs and case management don't actually remove all transmission, right? They work to a certain extent, but not all transmission. And that is why that's another gap in protection. And that is why we need vector control. And we have a bunch of innovative interventions coming up, and I think other speakers are going to be talking about them. So overall, I think vector control will remain absolutely central over the next decade. But I don't think the future is simply going to be better versions of the same tools we already have. We have spent decades relying heavily on a few interventions, particularly nets and IRS for malaria, and we're increasingly seeing the limits of that approach. Resistance is part of it, but so is mosquito behavior, outdoor transmission, varying human behavior. Climate change is messing things up. Urbanization and the fact that transmission is incredibly heterogeneous, even across a city or a village. Transmission can change from point to point. There are genuinely exciting new tools coming through from SEs, spatial emitters, and new chemistries to Wolbachia and insecticides and genetic approaches, and much better ways of targeting interventions as well. But I don't think there will be one disruptive technology that suddenly solves everything. For me, the real disruption, how we can change our mindsets, is how we use vector control. I think we'll move away, and I hope we'll move away from asking what intervention should everyone get, and re-ask and ask ourselves, where is transmission actually happening? Who is being exposed? And what combination of tools actually interrupts it right there, right? It could be different things based on different places and transmission spaces. So the future is probably not one magic bullet. It's probably going to be better surveillance, better targeting, more diverse tools that combat different gaps in protection, and much more intelligent combinations of them. Yeah. Thank you very much.
Yeah, so Neil, what a wonderful presentation in your usual very clear, calm way, explaining, this new WHO endorsed product. So thank you for explaining that to us. And for your words at the end, the speculating, not speculating, but giving us your impressions about, the remaining importance of vector control, but not necessarily over relying on one or two specific ones. Very appropriate messaging there. Thank you very much, Neil. We're going to move on to Professor Carlos Shakur, and he will be talking about the potential of insecticides in vector control. Carlos is a medical doctor with a separate PhD as well, originally from Venezuela, but he's now based at the University of Navarra in Spain. And his research focuses on the use of ivermectin and other insecticides for the integrated control of malaria and neglected tropical diseases. He's particularly interested in a One Health approach to malaria control, and he has a huge amount of experience, field experience in the Amazon region, Mozambique, Kenya, Tanzania, Uganda, and also Democratic Republic of Congo. So we look forward to your presentation, Carlos, and we're grateful for you being able and willing to join us and share your experiences. Over to you, please, Carlos.
Thank you. Can you see this clearly?
Yes, now we can, absolutely.
All right, so thank you, everyone. And Leo asked me to talk about insecticides, but also asked me to put an emphasis on the One Health approach. So I'll do that. And Wait a second, my slides seem to be all right. So what I'll do is I'll briefly mention residual transmission, and then I'll talk about the key role of livestock and livestock feeding vectors. Then I'll talk about ivermectin. I'll present the results of Bohemia, and then touch on the livestock part of Bohemia, and then talk about what opportunities we have with livestock and endectocides. So residual transmission has been mentioned already, and basically we have become really good at targeting mosquitoes that feed indoors and at night. But if they feed outdoors or if they feed outside sleeping hours, then we have a gap, as Neil mentioned. And then we also have a gap on mosquitoes that feed sometimes in livestock and then come back to feed on humans. And this livestock blood becomes a free meal that is normally free of bed nets or insecticides. So that allows mosquitoes to thrive, and I'll touch on that. So endectocides are an idea to target these gaps. So why are livestock and livestock feeding vectors a key problem? We touched on this on a paper that we published years ago with Jerry and all these people mentioned here in the BMJ Global Health, talking about why there is need to go beyond personal protection. And there, one of the things we mentioned is that the best vectors in Africa is Gambi and Finestus. If you look at the x-axis here, that's the proportion of blood meals taken from humans. So the best vectors feed mostly on humans, over 80% of blood meals on humans. And that makes them the best vectors because they're constantly feeding on blood that contains malaria. Animal blood does not contain malaria. But if you look at these other vectors like Arabiensis, then they're all over the place. Sometimes they feed 80% on humans, sometimes they feed 5% on human. So that's a problem because no single intervention will really tackle them. In Asia, the examples would be Faroti and Stevensii, which also have broad a range of blood meals taken from humans or animals. Then if you look at the numbers of mosquitoes, and if you look down here in my screen, there's these two triangles, right? On the left of the screen, there's the more zoophagic mosquitoes, and on the right of the screen, there's the more anthropophagic mosquitoes. So those that are purely human feeders are few number of species, but those that are often feeding on cattle, but occasionally feeding on humans, then there's a much larger number of species. And that's a problem because of their sheer numbers. Even if their blood meals are just occasionally taken from humans, this large number of mosquitoes allows for transmission to continue, even if the human biters are very well covered. And yet another problem here is that you see black, gray, and white. Black is high capacity vectors. Gray is this so-known low capacity vectors that I mentioned that feed occasionally on humans and animals. And those tend to bite at dusk or dawn. So now we add the dimension of time as well. So it's not just biting outdoors, it's also biting outside sleeping hours. It's also biting when people are already out of the house and doing field work. So these added gaps continue to sustain malaria transmission despite the current vector control tools. So ivermectin, this is my work from 16 years ago, in which we showed that mosquitoes fed on ivermectin treated people in black die much faster than mosquitoes treated on non-ivermectin treated people in the dotted line. This has been repeated over the years by people who are much better entomologists than I was in 2010 and actually got a proper result showing that the gap is actually much bigger when you don't have an MD taking control of your mosquito colonies, and then the control group survives as they should be. This is concentration dependent. So you want to have a high dose of ivermectin probably repeated over time, or you want to have a long-lasting formulation so you can have this mortality effect sustain on the mosquitoes for a longer period of time. In Bohemia, we actually tried to do that. That was a large consortium that took place from 2019 to 2025. And it consisted in all this group. It was funded by Unitaid. There's no time to mention all the key people that worked on this, but I honor them by showing their images in this slide. We had a great secretariat and senior people. And at some point, we had over 400 people working in the project, including field workers. So we wanted to generate quality data to have an evidence-based decision by the WHO on the use of ivermectin or endectocides for malaria control. In Kenya, we had a cluster randomized trial that had 84 clusters, and we gave humans 400 micrograms of kilo of ivermectin in a single dose once a month for 3 continuous months right at the start of the rainy season. And then in each cluster, we followed the children that were 5 to 15 for 6 months to see what happened to their malaria incidence. It is important to know that there was an 80% ITN ownership and an 80% usage of these bed nets. Also, 2/3 of the homes had at least one bed net for every 2 people. So this is a district that has, I would say, ITNs at capacity. They're well distributed, people are using them, and there's universal access. And yet, at baseline, the prevalence of malaria in children 5 to 15 was about 30%. So despite this great usage of bed nets, we still had a very high prevalence of malaria at baseline. We included almost 29,000 participants. The criteria to receive the drug was being non-pregnant and weighing more than 15 kilos. And what we see is that there's a reduction of the malaria incidence in the group that took ivermectin. And this reduction was actually 26%. So, and that's on top of bed nets. So despite this great bed net penetration and usage, we still managed to reduce by 26% malaria incidence on top of that by just giving 3 doses of drug once a month for 3 months. And that's what I said, is a 26% reduction. And not only that, but the community was very happy about the effect on bedbugs because they noticed that bedbugs were dying very quickly in the social science team. So we went and tested it on the bedbug colony we had, and the concentrations we achieved, actually bedbugs died very quickly. Not only that, but in the Mozambique trial, we had communities with a 13% prevalence of scabies at baseline. And what we saw is that there was a more than 80% reduction in the ivermectin clusters at the end of the trial. And interestingly, this reduction happened both in people who received the drug, but also in people who were not eligible for treatment, like pregnant women or children under 5, or people who just refused to take the drug. They also saw the rates of scabies drop. And that's because there's a general drop in scabies in the whole population. A similar result was found in head lice. Sorry to say, if you want to have a reduction in your head lice, you do need to take the drug. It doesn't matter if your neighbor does it or your parents do. There's not indirect benefit here, as we saw in scabies. So what we see here is an excellent opportunity for the integrated control of malaria, scabies, onco, LF, head lice, strongyloides, and bedbugs. Maybe some other helminth that would fuel acceptability, because people don't realize the malaria benefit right away, but they do realize the scabies and the parasites and the skin parasites benefits right away, and that has efficiency and cost savings. So what did we do about livestock? Well, in Mozambique, we did have an arm in which we treated all the humans and all the livestock, that is cattle or pigs, in the cluster. But in Mozambique, we also had an unexpected visit by Cyclone Gombi, And Gombe actually flooded the whole district where we were working in Mopeya and really made operations very difficult. Not only operations, but also distribution and access. And also it killed about 60 to 80% of all the pigs in the area, which was our target livestock there. So displaced population, flooded areas, working with canoes, and of course cholera didn't really help. So the results in Mozambique, and that's the bad news, could not be really interpreted appropriately because the coverage of population and the starting of the trial was so severely damaged by this. So the bad news is that we don't have a second trial that is as Neil mentioned, needed for WHO to make a decision, but we also don't have a trial that included livestock because the Kenya one did not. These results were published both in the New England Journal of Medicine and eClinical Medicine from Mozambique, but we did manage to do some livestock work. And in Kenya, Almudena and Rachel did a great work looking at cattle density and ownership and movement. And what we see is that if you look at the below the number letter B here, you see that for a single house, we mapped it as a red dot. And then we looked at where cattle are around in different concentric circles. And normally people look at cattle as a variable in malaria in which they ask, well, do you own cattle or not? We went beyond that. And it's not just if you own cattle, but what about your neighbors and where is this cattle located? And that's what you see in A above. Like we wanted to map the density of cattle. We wanted to map the human to cattle density, and we wanted to look at the ratio. And what we see is that cattle that is close to you doesn't seem to affect your malaria rates. Perhaps that's a sign of zooprophylaxis. The mosquitoes are being driven to the cattle and away from the houses. But cattle that live around 400 or 600 meters, which is the average flying range in Kenya of anophelines, do increase your malaria risk by an odds ratio of 2. And that happens with cattle density, that happens with cattle to human ratio. So not only that, but there was this question, well, what happens with cattle that moves around? So we tagged cows in different herds with GPS collar that mark their position every minute. And what we see is that cattle actually do roams during the day, but they sleep at the home at night. And that's been demonstrated in our study. So if mosquitoes are feeding on them and they tend to do so at dusk or dawn or during the night, then the cattle will be around. So there's not, so we think this will help plan for future studies around the role of livestock and cattle, and hopefully the role of livestock insecticides and cattle. So I always show this slide talking about ivermectin, and if you want to expand this efficacy, well, you need to have higher plasma levels, right? I mentioned that before. The higher the dose, then the better the efficacy would be. Then you need to sustain those levels, and that means you can give different doses over the rainy season, or you can use a long-lasting injectable, which is one of the things that is being developed right now. And the other one is coverage. The more people that are covered, the better that this will result. But the alternative is to look at this as blood coverage. So the more blood sources are covered, the better. So ideally, the next trials will incorporate, again, human and livestock treatment so that you would leave the mosquitoes the least amount of available options. Challenges and opportunities, I'm almost there. Basically, one of the challenges with cattle is withdrawal time, right? If you treat an animal with a drug, be it an insecticide or antibiotic or any other thing, you need to wait a few days, sometimes weeks, before you can milk or slaughter them. And that creates some challenges, but there's solutions for that, such as refugia or marking. There's also the challenge of potential fueling resistance in veterinary helminths, which are the key target of insecticides. And again, refugia, mosaics, long-lasting formulations may help with that. And then there's this challenge of potential selection for anthropophagic vectors. That is, if you do kill a lot of the mosquitoes feeding on cattle, well, would that allow this ecological niche to be occupied by more mosquitoes that feed more on humans? Then opportunities, there's long-lasting formulations being developed both for humans and livestock usage. Livestock provides the opportunity for mosaics. You can use an endectocide in humans, you can use a different one in animals, and you can use different ones and create mosaics. If you treat animals early on, if you treat calves early on, they gain more weight. If you treat cows, they produce more milk. So that has food safety and indirect economic gains, which is something often overlooked. And these economic gains can be reinvested in the health of the family. And then there's also refugia, in which it's an opportunity to manage resistance proactively. So there's people working already in long-lasting formulations of ivermectin and other endectocides for cattle or livestock. There's the Lima trial happening in Kenya soon. So there's ongoing work, and yes, we missed the target of providing WHO with 2 trials, but there's ongoing work, and I'm hopeful this will result in a success that would increase the arsenal we need to target all those gaps Neil was mentioning. And this is what I said, at some point we had a huge group working with us, and we're very grateful for all their work. So thank you very much.
Carlos, thank you so much for explaining your work on ivermectin and the principles of endectocides and so on. I have a particularly soft spot for endectocides, especially its potential role here in the Greater Mekong subregion and many other countries in Asia Pacific, where many rural households have several cattle closely associated with their households. They sleep underneath houses on stilts. In India and some other countries, households even have cattle inside the house. So, well, how much better can that get when you offer household owners a product that can simultaneously deal with the worm burden, reducing helminth prevalence, and also a deal with malaria reduction, especially in a residual malaria situation along border areas or rural areas with persisting malaria at low transmission rates. I think endectocides have a particularly useful role to contribute. So thank you, Carlos. A wonderful presentation. We're going to move on to Professor Jackson Champer to share insights on gene drive. Professor Jackson Champer is originally from New York City. He received a Bachelor of Science in Physics and Mathematics from the University of Oregon, and then a Master of Science in Physics from UCLA. And then he switched his focus to biology, receiving a PhD in 2015. And after a postdoctoral fellowship at Cornell University, he opened his lab at the Beijing Peking University in May 2021. And then since then, Jackson has been an assistant professor in the Center for Bioinformatics, and his lab focuses on experiments and modeling of gene drive and related areas of genetic population engineering. We look forward to your presentation. Thank you for being willing to join our webinar. Over to you, Jackson.
Yeah, thanks for inviting me. Let me share my screen here. All right, so my lab is all about gene drive, and we are especially focused on using gene drive for malaria control. So first of all, what is a gene drive? They're both natural and engineered gene drives. So these things are everywhere already in the wild, but engineered gene drives have not yet been released, and that's what we work on. The idea is that we make some individuals, say mosquitoes, with a gene drive, then we release them into a population, and over time, the gene drive will spread to the whole population. It works by biasing inheritance. So a normal allele in a heterozygote, one allele, passes on that allele to just half of the offspring. The other half don't get it. A gene drive, though, even if you have just one copy of gene drive in your genome, you can pass it on to all of the offspring. And that biased inheritance is what allows a gene drive to spread through the population. And all it takes is a few generations. So if you assume that these generations are mosquito generations, each one is maybe only a few weeks. The gene drive can go from 5% to 100% in potentially just a year. So there's two main things we can do with these gene drives. The first is modification. We spread the gene drive to the whole population, and then that gene drive is going to do something we want, like preventing malaria from being transmitted. We can also configure the gene drive for population suppression. We'll talk more about that soon. So, of course, the most important use for gene drives would be fighting against malaria. There are already a few different gene drives tested in different malaria species, and there are cargo genes which can prevent these malaria mosquitoes from transmitting malaria. But I do want to briefly mention that we can use gene drives for other mosquitoes and vector-borne diseases as well, and even various invasive species and agricultural pests to control all these in a more environmentally friendly way. So how do these gene drives actually work? Most of the modern gene drives use CRISPR-Cas9. So the gene drive has CRISPR-Cas9, and that cleaves the wild type allele on the other chromosome. So you've got two chromosomes, one gene drive, one wild type. The gene drive cuts the wild type chromosome. And then on either side of the wild type chromosome, the DNA is identical to the DNA on either side of the gene drive in the gene drive chromosome. This then can result in homology-directed repair, where the cell uses the gene drive as a template for that repair, copying it to the other chromosome. That's why, even though most cells of a gene drive organism might just be heterozygotes for the gene drive, one gene drive, in the germline reproductive cells, the cells that get passed on to the next generation, they can be homozygotes. And that is how the gene drive spreads through the population. But these CRISPR homing gene drives have been around for over 10 years, and they have still not been released. The question is why? Well, there's actually some major technical challenges that need to be overcome before a gene drive is actually good enough to release. And one of the big challenges is resistance. So if we don't repair this DNA break by homology-directed repair, if we repair it by end-joining repair instead, that just stitches together these two ends and often changes the sequence by a complicated molecular mechanism. That sequence can then no longer be recognized by the gene drive allele. It can no longer be cleaved. And that means that it can never be converted to a gene drive. Resistance alleles that have a fitness advantage over the gene drive, which we sometimes call R1 alleles or functional resistance alleles, are the big problem. If we have a modification drive that is super efficient, even gets to 99%, if we get even a few of these functional resistance alleles, then over time, they can take over in the population. For a population suppression drive, it's even worse. Those functional resistance alleles will increase almost as quickly as the gene drive did, resulting in immediate suppression drive failure. So how can we potentially overcome this challenge of resistance alleles? Our first attempt here was in Anopheles stevensii. These are malaria mosquitoes. They don't transmit quite as much malaria as the Anopheles gambii complex, but they're still pretty bad. They're native in South Asia, but quite invasive all over Africa. And this is because these Anopheles stevensii mosquitoes can live in urban environments where there's more and more people living. To make a suppression drive in Anopheles stevensii, we started with a similar design that other groups have been working on in Anopheles gambii, but we tried to make two improvements. The first was upgrading the Cas9 promoter to a promoter that may have worked a little better in Anopheles gambii. The second was using two guide RNAs, and here we're targeting the female-specific exon of double sex. So that's basically like targeting a female fertility gene. The idea is that we spread the gene drive through the population, and pretty soon all the females are gene drive homozygotes. They're sterile, so that means they won't bite and they won't be able to reproduce at all, thus eventually eliminating the population. But here we encountered another gene drive challenge. Even though these gRNAs should be enough to prevent functional resistance, at least in the lab, this gene drive simply wasn't strong enough. Only 3/4 of individuals inherited the gene drive when their parent was a heterozygote, regardless of whether that gene drive parent was female or male. So this means that we still have a gene drive. It's still a lot more than 50%. But because this is a suppression drive, it won't have enough power to suppress the population. We need much higher inheritance if we want to actually suppress a population rather than just modifying it. And the reason we got lower efficiency is because even though this nanos promoter works really well in Anopheles gambii, it did not work well in Anopheles stevensii. So even between different closely related species, you can sometimes still see some big differences in gene drive performance, even if the gene drive is similar. So we replaced it with the ZPG promoter, which works well in Anopheles gambii. And now we've got that really high inheritance that can give us a potentially strong gene drive. We did encounter another small problem in that it's not just the female gene drive homozygotes with two copies of the gene drive that are sterile. The males were too, another difference from Anopheles gambii. So we're working on new gene drives to overcome this, but we still wanted to test this gene drive because it had very high inheritance. So we released it into a cage population at low frequency. And in each generation, we tracked the frequency of the gene drive in these cages. Now, the gene drive has reached high frequency in 3 of the cages, and we've started to count the number of eggs that these female mosquitoes are laying. And one of them has now fell almost to 0, and the second one is definitely heading in that direction. So even though this gene drive has these higher fitness costs and is going to be a lot slower, it might still be able to work, at least in laboratory populations. So that's certainly good progress in Anopheles stevensii. And other groups have made suppression drives in Anopheles gambii that also work quite well and might be getting pretty close to being ready for release. But these homing gene drives, whether they're for modification or suppression, tend to spread very widely. This may be what we want, but in some areas, it might be difficult to get socio-political permission to release a gene drive that can potentially spread everywhere. And of course, when dealing with non-vectors, like the invasive species or agricultural pests, we might then also need to keep the gene drive confined to a target population. To do that, we're going to need a different type of gene drive. But there are actually many, many different types of gene drives with many different properties. Some of those properties can give us confinement. For these gene drives, you need to release a little more at first, because if you release below their threshold, they'll just go away over time, unlike the 0 threshold drives, which can spread everywhere from even a small release. But for these threshold-based drives, if we release enough, which can still be a pretty small number, over time, you see that they're spreading in this left population. But the migration to the right population is so small that the gene drive will never spread significantly to that population. So it's confined in space. We can also have self-limiting gene drives, which might spread at first, but then they go away. These gene drives are confined in time. The simplest kind of threshold-based CRISPR gene drive is the TARE drive. We made an initial version of this one in Anopheles stevensii, and it's a little bit more complicated molecularly than the suppression drive. You got to insert it in one part of the gene, and then your CRISPR targets a later part of the gene. So this gene drive doesn't copy itself. It breaks the target gene, and then these target broken genes are removed from the population. This leaves only the gene drive, which has a working copy of the target gene. So rather than copying itself, this gene drive works by getting rid of the competition. So this means that we have to release a lot in order for this gene drive to then spread in the population. In this first try, the gene drive efficiency was a little on the low side, and we got an unexpected source of functional resistance. But we think that with some improved designs that we're already working on in the laboratory, we might be able to make a good version of this that can spread more strongly without resistance. We're also working on homing rescue drives, which can carry some anti-malarial cargoes and also spread in a population to modify it rather than suppress it. And finally, I want to talk about self-limiting suppression drives. The idea behind this is that we release the drive in this gray shaded region. And for self-limiting systems, we still have to release a lot more drive than for the self-sustaining gene drives. But when we release it, the population goes down and the frequency of the gene drive goes up. But for the suppression self-limiting drives, as soon as we stop releasing over here, we can see that the drive frequency is going down again, and pretty soon the population recovers. So it's a temporary gene drive. Even though we need to release a lot more of this, it's still better than state-of-the-art techniques such as SIT and RIDL. So SIT is basically just releasing lots of sterile males. They mate with the females, and then the females will have no offspring. So the population is lower in the next generation. But for this, even when this system works perfectly, in our model, we've got to release 4.3 males for every male in the population. And we've got to do this every generation. That's a lot of mosquitoes. But if we have these self-limiting suppression drives, then if the drive efficiency is high enough, we can release a far smaller number of mosquitoes to eliminate the target population. And when we tried that in split form for Anopheles stevensii, we found that the males with this gene drive will spread it to 100% of their offspring. So, that's in this far dot over here. It might be 10 or even 50 times less mosquitoes than sterile insect technique, which is not strong enough to deal with most malaria mosquitoes. And all the females that had this version of the gene drive were completely sterile. So it had exactly the properties we want for a strong self-limiting drive. We just have to put the components together into a single system now, and then we might have a type of self-limiting gene drive that can suppress mosquitoes in cities, but will then not spread outside of the release area. All right, that's my whole presentation. I'd like to quickly thank my lab members, which collected most of the data that you saw there. And just to offer my thoughts for the future of malaria control going forward, I want to point out that these gene drives could be extremely powerful with a very low resource need, especially the self-sustaining homing drives. But so far, there have not been gene drive releases. They might be coming quite soon for Anopheles gambii complex mosquitoes and Anopheles stevensii mosquitoes, and maybe they can solve a big part of the problem. But they are still ultimately untested, and we'll need to see how they perform in the field before we know how useful they'll be for vector control as a whole. The other issue is that existing vector technologies can often be effective against a broad spectrum of vectors. Gene drives are very species specific. So we're doing a great job with Anopheles gambii and stevensii, but other important Anopheles species, especially those in Asia and Pacific Islands, research has only just started for these. So these might be a little further off. We believe that in Anopheles mosquitoes, at least, we can get gene drives working very well. But it still does often take a few years of research to get these, even after we know how to overcome all these technical challenges. And in other mosquito vectors, it's even harder to make a really good gene drive. So it has the potential for very huge impact, but it absolutely cannot replace existing systems, which are all we have to work with right now.
Is that it, Jackson?
Yes, that's all I have.
All right.
Thanks very much.
Yeah, no, well, from us to you, thank you so much, Jackson. What a wonderful presentation. I hope our audience members are thoroughly enjoying these presentations and learning as much as I am. And certainly yours was a very, very engaging, informative presentation. Thank you so much, Jackson. And you made some cautionary statements. There are, it's not a one size fits all. It's very narrowly targeted to specific species. Many people would say that's a good thing. But there's a way to go yet. Know that there are a lot of people that want you to succeed and are quietly, fully waving the flag for you guys and wishing you well. So, from the drug perspective, vaccines, there's a strong hope for the future of vaccines in really making a difference in malaria burden reduction. And a lot of us are also quietly thinking that maybe gene drive ultimately, when it's ready, can make a massive impact as well if we target particular species responsible for a high transmission. Thank you, Jackson. Thank you sincerely. So we move on to our next presentation. I'd like to tell our audience members there are more than 560 of you that are currently linked in, and it shows you the level of interest that this particular webinar has generated. Wonderful to see such interest levels. And I know we're stretching your patience by having so many presentations, but like me, I'm hoping you're enjoying it as much as we all are. So we're going to move on to Dr. Claudia Surjajaja, who will share with us some of the impressive work that has been done using Wolbachia for the control of Aedes-borne arboviruses, especially dengue. So Claudia is a public health professional with over 30 years of experience across more than 30 countries, working with a variety of international organizations. Claudia is passionate about global health, the design, management, and evaluation of health programs, and health system strengthening. Claudia collects degrees like few others, and her qualifications include Doctor of Dental Surgery, Master of Public Health in Health Service Management, Master of Science in Medical Ethics, Doctor of Public Health in Epidemiology and Population Health from London School of Hygiene and Tropical Medicine. The list goes on, but let me stop there. Currently, Dr. Surjajaja serves as Director for Advocacy and External Relations Asia at the World Mosquito Program at Monash University. And she's also the Executive Director for the Alert Asia Foundation. So Claudia, we look forward to your presentation. Thank you for your time and effort. Over to you, Claudia.
Thank you. Good afternoon, everyone, and thank you for having me. So when we talk about mosquitoes, we usually talk about them as the enemy, something we need to kill, control, or eliminate. But what if instead of fighting mosquitoes, we could turn them into our allies? That is actually the idea behind Wolbachia. Today, however, I don't want to focus only on the science. The science is fascinating and the evidence is strong. But what I'd like to share are some of the lessons we have learned from taking that science out of the laboratory and turning it into real public health impact. Because after years of working with Wolbachia across very different countries and communities, one thing has become very clear to us. The breakthrough science is only the beginning. And that is what I'd like to talk about today, what comes after the breakthrough. Next slide, please. So what is the World Mosquito Program? The World Mosquito Program is not for profit group owned by Monash University that works to protect the global community. So we particularly work in Aedes aegypti. So to prevent dengue transmitted by Aedes aegypti, such as dengue, Zika, yellow fever, and chikungunya. And we use a natural occurring bacteria called Wolbachia to reduce the ability of mosquitoes to transmit viruses to humans. Next, please. So how does it work? So WMP transferred Wolbachia from the fruit fly, this is 15 years ago, into Aedes aegypti mosquitoes. And we have proof that this is safe for human, animals, and also the environment. We already work now in 16 countries globally. And this is not genetically modified. So this is not non-GMO product. This is natural occur Wolbachia in the nature. Like 60% of the insects have Wolbachia. So we introduce Wolbachia into Aedes aegypti, and this is the cell. And then it works to all 4 serotypes of dengue, and then also the other like disease that I already mentioned. Next, please. Next slide, please. So how does it work? So this is the and this black mosquito as the mosquitoes with Wolbachia. This is the blue one here on the right. Are released into the wild mosquito population this 10 months. And once a threshold frequency of Wolbachia mosquitoes, usually like more than 80%, reach Wolbachia will continue to spread because they're mating with the wild mosquitoes. Next, please. Next slide, please. Next slide, please.
So we're in here into the plasmid incompatibility.
Yeah.
Yep.
Right. I just see it right now. All right. So how does it work? So this is what happened when the blue one, again, the blue one is the mosquito with Wolbachia, and the black one is a non-Wolbachia mosquito. So in A is when a male mosquito with Wolbachia mate with the wild female mosquito, the eggs will not hatch. And then the B model is when the wild mosquito, male wild mosquito, mate with the female mosquitoes with Wolbachia. All the offspring will have Wolbachia. The same with C. If both mosquitoes, female and male, have Wolbachia, and all the offsprings will have Wolbachia. So that's why this is a sustainable approach. Next, please. So how does it work? So again, this is how we work with the community. Usually we have the preparation phase before this, and then to train people. And then when we start the release, which is 3 to 6 months, and there are many methods of release. We can release the eggs, we can release the larvae and adult mosquitoes. And then they're mating like in the previous slide. And then when the threshold is reached, and then we move to other side. We were no longer there because it will spread automatically or naturally. And then, of course, the community is protected from this is long-term without the need of more releases. So one release and that's it. Next, please. And this is our impact. Currently, we work in 16 countries across four different continents. It's Latin America, Asia, Pacific, and the new one is Africa. And then another 2 countries also using Wolbachia, that is Singapore and Malaysia, but it's not part of WMP or World Mosquito Program. Next, please. And then this is the clinical trial in Yogyakarta, so to show also the efficacy of this method. And in Yogyakarta, after 5 years of post-release, we monitor it back, and then the Wolbachia remains at 85%, and then it covers about 400,000 people. And in Niterói, Brazil, it's the same thing. Wolbachia keeps stable 5 years after release at the threshold of more than 90%. The same with Medellin in Colombia. We cover 2.3 million people. After 4 years of post-release, Wolbachia remains there at above 80%. Next, please. And next, please. And now, what about the dengue incidence reduced? So these are the sample of the 16 countries we work in Cairns. Cairns is in Australia, where we work first time to release Wolbachia about more than 12 years ago. And then we came back after 10 years even, and then there is no single local infection in dengue infection. And after almost 130 months of post-release, Wolbachia remains 98%. And the same with Townsville and Yogyakarta, Vitlong in Vietnam, Rio de Janeiro, Niterói, and other places. So Wolbachia remains stable after years, after post-release. Next, please. Yes, and then this Wolbachia, and we use WML. It's already approved or evidence provided. It's already approved by the feedback control group, factor control group of WHO. And currently, WHO is still developing the guideline and also the PQ. Next, please. Yes, now, what about Wolbachia in Anopheles? Because we're only talking about ITIS. I try to summarize all the research out there. WMP is not in this research in Anopheles, and I will tell you why later on. But this is all the examples or the work by our colleagues out in introducing Wolbachia in Anopheles research. Next, please. Yeah, so the summary. So what is the possibility of using Wolbachia for Anopheles control? So from those studies, introduction to Anopheles GmbH using thoracic injection, so no heritable injections. And based on the publication that infections are still unstable and transient. And sometimes we see parasite blocking, sometimes it's just an enhancement. So at the moment, there is no stable maternally inherited laboratory line has been created. So no candidate for Wolbachia, both suppression and replacement method for Anopheles GmbH. And then for Anopheles stephensi, and then show stable infections, but also still limited evidence on parasite blocking. It is potential candidate, but still long way to go before operational in urban areas. Very challenging, especially when the population becomes more rural. So for population replacement, there's also only one candidate with both demonstrated stable inheritance and parasite blocking. But however, again, remains a laboratory proof of principle. The journey for stable infection to scale release is still a long one. And then another one is because now the GM or genetic modifying Anopheles research now outpacing the Wolbachia Anopheles research due to Anopheles ecology and biology differences. And then this is the most challenging is the operational implementation, because with the current release technology, it will be harder than Aedes aegypti, because Anopheles again has different biology and ecology, and usually it's highly abundant in nature, largely rural, and then capable to dispersing in substantial distance compared to aegypti. So it will need enormous numbers of mosquitoes released over very large areas. Next, please. So what is the future of Wolbachia for expanded Aedes control? So with Aedes aegypti, we have already a strong scientific foundation. Wolbachia is stable in mosquito populations, substantially reduced ability to transmit the disease, dengue and other 3 diseases, and maybe another arbovirus. But evidence of sustained disease reduction is really strong at population scale. So this already shows greatest public health impact. And our priority is to maximize public health impact through deployable approach at scale. So from pilot sites to national level in the country. So take the evidence and capability, focus on the enormous and growing burden of dengue and other Aedes-borne disease, and take an intervention that we know works and make it available at much greater scale. Next, please. So what is the lessons? So lesson from Wolbachia for future mosquito vector control. So Wolbachia, sorry. So Wolbachia has taught us that breakthrough science, as I mentioned before, is really only the beginning. Again, the breakthrough science is only the beginning. So getting from the laboratory to public health impact requires much more. It needs regulations, financing, production, technical capability, partnership, and above all, we need support from the community. But I think there is also an even bigger lesson here for the future of vector control. We should not measure success by how long countries depend to organizations like ours. The measure of success is actually the point where the countries can do it by themselves, has knowledge, system, and capability to do it by themselves. Because ultimately, as mentioned, sustainable impact comes from ownership. So when knowledge and capability sit within the countries and the communities, an innovation stops being a project and it becomes a public health system in the country and hopefully worldwide. So if there is one thought I would leave with you, is this. Again, science can change what is possible, redefine possible, but ownership is what makes change last. And that is the lesson that goes far beyond Wolbachia. Thank you.
What a wonderful message for all of us to take to heart, that we need to take ownership, personal ownership, not rely on governments, top-down approach. We need to adopt and try and help ourselves as best possible. So, Claudia, thank you for sharing with us the great successes and value that Wolbachia has brought to AIDS control. You know, I don't think many people, well, let me put it this way. Malaria is strongly declining in large parts of Asia Pacific, but dengue is on the increase. And the bulk of the world's malaria cases are here in Asia Pacific. And so, you know, dengue is going to be the next malaria in Asia Pacific. And it's very reassuring to know that there are approaches such as Wolbachia that can have such an incisive impact on dengue vectors. My only concern is I get the impression, I may be wrong, it's expensive, millions of dollars, to treat like a suburb, a section of a city. So, you know, where does the money come from to spread it out across an entire country? But, you know, that's subject for another talk maybe sometime. Unless you want to answer very quickly, Claudia, can one apply Wolbachia? Yeah.
I'm sorry, it's really noisy here. I try to answer quickly. Again, this is one stop, actually one release only. So it's heavy upfront, but the more you release, the more site, because it's after we release and Wolbachia is stable, we move to other site. So it's costly upfront. But the more we add the sites, it's getting cheaper and cheaper. Actually, it's about, if I'm not mistaken, less than $1 per person.
That's very reassuring. Thank you for that additional information, Claudia. So our next and final presentation will be by Dr. Mohammed Mukhtar, who will share with us his thoughts around the interplay and the importance of partnerships between control programs, academia, and industry. Very, very important things to discuss. Dr. Mukhtar is director of the Pakistan Directorate of Malaria Control and Other Vector-Borne Diseases, and he's the author of multiple national policy and strategic guidelines, including on climate change, in public health, vector control, in humanitarian settings, such as the huge floods that Pakistan was subjected to in 2022. Lots more high value products that he's produced, highly productive, and a thought leader for innovative approaches to vector control. Dr. Mukhtar also happens to be the chair of our APMEN Vector Control Working Group, and a very active one at that. So thank you, Dr. Mukhtar. Over to you.
Hi, everyone. Can you see my slide? Is it
Not yet.
Oh.
If you can go full screen, please, Dr. Mukhtar.
Yeah. It's okay?
No, not yet. If you can, there we go. Wonderful.
Yeah.
Thank you, Dr. Mukhtar. Please go ahead.
Thank you. Thank you very much. My presentation will actually just summarizes what my previous colleagues, very esteemed colleagues, have presented. That the current vector control tools are not sufficient in the scenario of the outdoor biting resistance development. So my presentation is to highlight the need of the innovations. Need of the innovation, not by a one actor, because actually what I'm going to talk, we are the three actors: program, academia, and industry. When these three actors join hand together, they can build some resilient future vector control new tools in the scenario of the challenges which I'm going to present. So my presentation mainly connecting the public health programs, academia, and industry to build the vector control toolbox for tomorrow. This is just the vector control changing scenario. You know that the vector ecology and transmission pattern has changed due to the climate change, one of the major drivers, and rapid and uncontrolled urbanization, and very important, vector adaptation to various climatic and ecological scenarios globally, and human behavior, sleeping activities like this. So these are these are the drivers which have changed the vector ecology and transmission pattern, and consequently outdoor biting is going on, and early evening and residual transmission, which my colleague already explained very well, are become increasingly important. And operational challenges are evolving faster than our existing tool. This is the key message. I have written other key message as well, that the operational challenges for the vector control in the changing landscape have become more faster than our existing tool. The future of transmission will not necessarily look like the past, not past even today. And our tool must evolve accordingly in keeping in view these challenges. Yes, we know that currently we have two core interventions: indoor residual spray and the use of ITN, and both interventions remain primarily indoor focused. And changing vector behavior, transmission pattern, which I already explained, reducing the efficacy of these conventional approaches. Outdoor biting, early evening, and residual transmission, difficult to address with these current tools. There is a gap in effective and practical and scalable tools for the outdoor transmission. What we need now, innovations that can complement the existing intervention and address these gaps. And what are the key messages from this slide which I want to communicate? That we cannot solve the tomorrow's vector control challenges with the yesterday's. Not I will put the today's toolbox. I will not focus the yesterday, but I will focus that even today's toolbox, what we have in our pocket for vector control, that are not sufficient based on these challenges which are evolving over the last 1 or 2 decades particularly. And what is the partnership? I use the term of the pro-actinet partnership. Well, you know that as beginning, I told there are three actors: national program, academia, and the industry. What the national program can do? I, as a national program manager of Pakistan, understand what is the challenges and what are the unmet needs currently for the program. Academia can bring the research, science, and evidence to address these needs. No program identify the challenges. Academia can address how we can generate some information through operational research. What are the needs? and industry can bring the innovation. Now you can see that program identified the challenges. Academia generate the knowledge that yes, there are challenges and how we can meet these challenges, and industry bring the innovation. So there are the triangle of the partnership, and what we can pro-accelerate, which indicate the program plus academia and industry. One partnership, three strengths: program, industry, and academia, and one goal, innovations. So one key message of by this partnership that the program define the problems, academia generate the knowledge and evidence, and industry translate into the solution. So you can see that the three actors can join hand together, then we can make some vector control tools for tomorrow's challenges. From now, how we can bridge the gap between the discovery and delivery. Now you can see that how we can make this gap, can bridge the gap between the discovery and delivery. Define the problem, developing the solution. Program, it is the job of the program. Generate the evidence and effectiveness, feasibility, and scalability. That is the academia role. Test the innovation in real-time program setting industry. So you can see, again, this is the same slide which I previously had, but in some more systematic way. And the key message, innovation only carried the impact when it reached to the field, which means that what we are doing, either as a program, either as academia, either as industry, it will be only benefited for the community when it will reach to the up to the community or target community where these innovation tools are needed. Either it's as a repellents or is a Wolbachia, very much interested. I'm extremely impressed by the gene drive Wolbachia today, because today I'm also learning a lot, because we are talking about the future vector control. What would be the future of the vector control? And I'm extremely impressed by the talks which my esteemed presenters have made this. So my is just the summarization of that how we can go for these innovations. What is the role of the program? What is the role of the academia? And what is the role of industry? When these three partners, actors join hand together, definitely we will build some brighter and healthier future through the vector control. Building the future. This is my final slide, because my present is very short. That again, the program, what is the role of the program? What is the academia? My key message is, if we want to build the vector control toolbox for tomorrow, we must start building the partnership of tomorrow today, which means that this is the right time when there are challenges are there from the field, from the industry, from the academia. And if we want to build a safer, healthier vector control intervention, we have the building the partnership today for tomorrow. I think this is my final message. Thank you very much for your kind attention.
Dr. Mukhtar, thank you as always for your insights and wisdom. So for those comments, thank you very much. Clearly, we must aim for a stronger collaboration between programs, control programs, and the research community, and then of course private sector, industry, and so on. So very important aspect to talk about. Thank you for that. So we're going to move on to our question and answer session. Thank you for your interest, our audience members, for your resilience in hanging in for so long. It's 1 and 3/4 hours already, and still there are well over 500 of you still linked in. Very impressive. Thank you for your sustained interest. So there's some questions that have been asked. I'm going to try and alternate between the different presenters so that each one gets a question. I see Jackson, unless I've missed it. Wulan, would you mind seeing if there's a question for Jackson? So I can also ask Jackson a question.
I think he already answered.
Oh, you already answered. Okay,
Yeah.
very good. Thank you, Jackson. Thank you for that. So there was
But there's another, I'm sorry to interrupt, but there's another new question here from Amir Javed from Pakistan.
Okay, keep that one in mind. When I get to Jackson's turn, I'll ask you to read it out. Thank you, Wulan. So I'm going to start with myself as having been the first speaker. There was a question here from Jeffrey He, who said, are we at risk of overstating the need for new technologies when the larger problem in many endemic countries is a failure to achieve adequate coverage, quality, and sustained implementation of interventions we already know work? And of course, that's a very valid statement, observation, question, absolutely relevant. And to emphasize that point, our existing tools, WHO recommended global fund supported tools of insecticide treated nets, indoor residual spraying, have been very effective in achieving elimination in more and more and more countries. They actually work. But the reality of the situation is that, you know, globally, malaria is actually increasing. So while there are these wonderful shining examples that, you know, we can reach out to and pin our hopes on, the reality is that you can take a horse to water, but you cannot force the horse to drink the water. You can hand out bed nets to millions of people, but you're only going to get 50, 60, 70% of those people actually using it. And then of course there's the added reality of increasingly remaining transmission is outside of where those existing WHO recommended tools are very effective. So, you know, biting takes place, as Neil very effectively pointed out, outside the hours of where people are protected by bed nets, especially outdoors. And we just don't have anything for that. So, you know, there is a need for integrated vector control. We cannot afford to lose ITNs and IRS. They remain critically important, but we need to supplement them. That's just my humble opinion, Jeffrey. So for Neil, as our next speaker in the lineup, there are some questions here. Neil, we get this question so frequently. The question is, why pre-qualification of spatial repellents, spatial emanators, and not full recommendation? So I think people get confused with that term, pre-qualification. Can you explain just a bit why not full recommendation and why pre-qualification and so on?
Yeah. So a recommendation is different from pre-qualification, and please correct me if I mess this up. So a pre-qualification is a product, a specific product that WHO, the entire WHO system, recommends for use and says you can use global funds to purchase this product for that paradigm for your country. But a recommendation is slightly different. A recommendation is where WHO says this paradigm, for example, spatial emanators, can be used for vector control. And to be able to say they recommend a specific paradigm, they need 2 randomized controlled trials demonstrating epidemiological impact. And at present, we just have one randomized controlled trial for dengue and one for malaria that demonstrated impact. So we would technically need another one. But because WHO, there's so much data with spatial emanators, WHO has decided on a conditional recommendations based on an overall review of all the data present. Now, with that conditional recommendation, now we can choose the products. And to have a product that is good enough to include with high levels of evidence that demonstrate that that we can include them in this paradigm for WHO or Global Fund use, we need pre-qualification. And that specific product has to go through very strict evaluation processes for WHO to say that this specific product, for example, Guardian, can be used within the spatial emanator paradigm that we have conditionally recommended. I hope that made sense.
Yeah, no, certainly. Thank you for dealing with that question. It's. Did you want to say something else, Neil?
No.
Okay. Yeah. It is a bit of a subtle difference, and many people just need a little bit of clarification, which you've done now. Thank you. And then it was Carlos who gave the next presentation. So, question for you, Carlos. As the ivermectin will pass through people and therefore be in human waste, do you know of any work that's been done on subsequent secondary ecological impacts, or even on flies and transmission of trachoma, maybe, but also for dung beetles feeding on waste products and so on? So, are you aware of any work that's been done on the kind of secondary effects of use of ivermectin?
Yes, indeed. Let me know if you hear me well, because I'm almost at the end of my battery.
Okay, so far very clear. Please proceed while you still have battery.
So
Thank you.
there is evidence that the residual ivermectin will have an effect on larvae that are breeding in areas where there's been contamination with human or cattle feces. And there's also some evidence that it would reduce the survival of larvae of other insects like flies. Importantly, it can also have an impact in non-target organisms like dung beetles, for example. So there has to be a strategy to manage that proactively if this is rolled out in humans or livestock, because you want to be careful with non-target organisms.
I think that answers exactly what the person wanted to know. So thank you for that, Carlos. Willa and Jackson was next in line. Do you have a question for Jackson that I missed?
Yeah, I think it's here. Wait a second. Trying to look correct. Just have to scroll it down.
I've got it. I can read it out if you want.
Okay,
Yeah.
okay, please.
Great.
Thank you, Jackson. Okay, cool.
So Amar Javed was interested in the TARE drive and whether there's a balance between gene drive efficiency and avoiding resistance alleles. And basically the answer to that is yes. And this is not just an issue for TARE drive. This is one of the engineering challenges that gene drive scientists have been trying to overcome for a while. If you put too much effort into dealing with resistance alleles, the efficiency of your gene drive, might go down so low that the gene drive still doesn't work that well. There are ways around this. Some of these ways are new techniques, like how we express the individual guide RNAs, exactly where do we cut. Other times it requires achieving a very fine balance. We need just the perfect level of expression. If we have less, then the gene drive efficiency will be low. If we have too much expression, there'll be so much Cas9 that even the eggs will have Cas9. And if the gene drive cuts in the eggs of the offspring, then we always get more resistance. So these challenges can vary greatly between different species. And fortunately, this is one of the reasons why Anopheles gene drives tend to work well. It's easier to overcome this balancing challenge in Anopheles than it is for other species, which are a little bit further behind in the development. There are solutions for all of these things, but some are easier than others.
Thank you for that, Jackson. Great comments. So here's a question for Claudia. Indonesia recorded, this is from Jeffrey here as well, Indonesia recorded more than 257,000 dengue cases and over 1,400 deaths in 2024. That's amazing. How should we reconcile strong local Wolbachia evidence with continuing high national dengue burden? And what does that tell us about the speed and scale required to produce a population level impact? I think it ties in with a question I asked you, Claudia, about implementing at scale and the costs associated with that. And you said it's front loaded. So once you've done the initial investment of setting up the warehouse scale facilities to churn out millions of mosquitoes, then you simply scale up and expand your releases. Is that the simple answer? And do you have examples where you've actually achieved that larger population scale implementation across a sizable proportion of any country? Has that been done with Wolbachia, please, Claudia?
Yeah, so as a background first, in Indonesia, the government already adopted, Ministry of Health already adopted, but it only has been done in 5 cities. And in 5 cities, it's not the whole cities. It's just like several sub-districts in the 5 cities. So that's still like a pilot in 5 cities. The example of national scale up is, of course, Brazil. So Brazil is now is like a lot than cities. I think they targeted more than 100 cities. That's why they produce a big production facility, can produce eggs, about 100 million eggs per week. And then just to give example how cost effective is that, may I share my screen? This is the example.
Of course, please go ahead. Please go ahead.
Yeah, so just to answer also which one is more costly, and that's not a vaccine, because again, Wolbachia is multivariant. It covers 4 diseases, so it's not just for dengue, but also yellow fever, Zika, and chikungunya. And now about this, can you see my screen?
Yes, we can. If you want to go full screen, then it becomes clearer.
I don't know how to do it.
Usually, I can't see the bottom of your screen, but it's usually bottom right, kind of near 80% along.
Okay.
Yeah, somewhere there.
Huh? Oh,
No, now it's gone. You've deleted it. Okay, bring it up as you did and leave it like, yeah.
So this is example of Bangladesh. And then as I mentioned before, it becomes cost effective after 5 years. And the more sites we adding is the more cost effective. And the cost scenario is for Bangladesh is $2 per person protected, but it can be less depending on the country's population and of course the CERs and then of the country and the GDP per country. So this is a sample of the scenario for $2 per person. So to averted, it's 5 years, and then we can see it's not cost effective at all. Compare with the ratio ICER to GDP. But in 10 years, we can see the decreased number of this, and it's already cost effective. And then it becomes only 2.887. And then after 20 years, it becomes lower and lower. So this is a scenario for Bangladesh, but again, in Brazil, it can be less than that because now it's full national scale up already. I hope that answered the question.
Yes, it does. Thank you for that, Claudia. That's very encouraging. After 5 years, you can bring the cost down very significantly. You just have to maintain the infrastructure and staffing levels. So, it can be a not self-perpetuating, but you can expand scale at a good, affordable cost, it seems like. So thank you for that. Very reassuring. Thank you, Claudia. So, Dr. Mukhtar, I'm going to make the last question to you. I don't see a particular question addressed to you. Maybe I've gone through ineffectively, but I don't see a question directed to you directly. So I'm going to ask you a random question, which did pop up earlier in the Q&A box, which is around the use of larval source management, which is given endorsement by WHO as a supplemental intervention. So, and WHO will only endorse larviciding, not larval source management as a whole. WHO only endorses as a supplementary method larviciding. But, you know, there's no money available for that. So if national programs want to use larviciding, then they would have to pay it out of domestic resources.
Yeah.
So a lot of people, there's a strong increasing adoption, shift back to larval source management and larviciding in Africa. Sorry, WHO only restricts that partial endorsement to those species that breed in few fixed and findable breeding habitats. And in Africa, that would basically be Anopheles arabiensis and maybe Anopheles gambiae sensu stricto as well. So, and it's the beauty of larval source management is you can get communities to participate in that. And also you can narrowly target it to peak dry season when breeding sites are very limited and population levels have shrunk. And, you know, you can really hit residual mosquito populations hard during the peak dry season, especially in Africa, where dry seasons are very pronounced in much of Africa. So there's a strong shift back to larval source management. Dr. Mukhtar, I wanted to ask you, does Pakistan use larval source management for Anopheles control? I know you do for Aedes. Do you want to say a few words about larval source management and larviciding maybe?
Thank you, Dr. Liu, for this wonderful question. It's one of my favorite questions to answer because the larval source management definitely WHO promote as a supplementary, not as a core intervention. A core intervention. You are absolutely right that currently Pakistan is using the larval source management for mainly the dengue control. For Anopheles, if we go back the history of the malaria control, vector control in Pakistan, till early 2000, we have a very big program of larval source management, larviciding for the Anopheles for malaria mosquitoes. But later on, when with the induction of the bed net to the Global Fund support, the program shift was only adulticide, like the mosquito nets, and to some extent indoor azure spray. But before 2000, we have one of the biggest larval source management or larviciding program in Asia when we are focusing the control of the malaria through using the larvicide for the Anopheles mosquito, Stephensii and Culissifaceae, which are the major vector. But currently we are limiting to for the dengue control because this is the for the dengue control. I evaluate the larval source management as a core intervention because for the dengue control, through the community engagement, through the community involvement, larval source management is the best intervention for dengue control, but it has some limitations for the malaria control, particularly after 2000 when we have a large-scale program of the bed net through the Global Fund. I think this is I have to make the clear.
No, thank you for that. Very clear, very useful response there. Thank you, Dr. Mukhtar. So I think we're overstaying our welcome. There's a sharp drop off in the audience number, which shows, you know, we've reached saturation or people have other responsibilities they need to get back to. You know, we've been at it for more than 2 hours. I personally have found it a highly stimulating and enjoyable 2 hours. And for that, on behalf of the audience and all of us at APMEN, A very deep, sincere thank you to our various speakers, expert presenters. Thank you for your wonderful presentations, words of wisdom, for the time and effort you put into this. Thank you so much. To our audience members, I want to ask if you could spend just a minute or two to respond to this poll quiz. It'll take you 1 minute, 2 minutes max. And it helps APMEN to understand, how you received this webinar. What, how useful did you find it? I'd also like to thank Dr. Porn and Wulan for staying with us and being our technical backstops, helping in the background, making the webinar possible from a technical support perspective. Thank you, Dr. Porn and Wulan. Again, to our speakers, thank you. Most importantly, to our audience members. What a wonderful audience. Such strong number of questions. Sorry that we couldn't get to all the questions. It's my fault. I scheduled too many speakers, but I hope you enjoyed it. So to all of you, thank you. See you next time.